Good afternoon, everyone. I am Ghazala Sabreen, PhD student under the guidance of Dr. Ruchi Gupta, Department of Hematology, SGPGIMS. I will be presenting my work on microRNA mediated regulation of Th17 and Treg cells in acquired aplastic anemia. In brief, acquired aplastic anemia is a bone marrow failure disease that is characterized by peripheral pancytopenia and hypocellular bone marrow. Pathogenesis of the disease includes intrinsic defect in the hematopoietic stem cells and the dysregulation in T cell homeostasis. The disease is majorly known to be immune mediated. There is alteration in T cell subsets along with transcriptional changes. This is a diagrammatic representation of the proposed mechanism of T cell mediated hematopoietic stem cell death in acquired aplastic anemia. Along with intrinsic defect in hematopoietic stem cells, it is well proven that T cells secrete various cytokines like interferon gamma and tumor necrosis factor alpha, which start a cascade of signaling in hematopoietic stem cells via the activation of Fas receptor through Fas ligand, which drive the cells towards apoptosis, leading to bone marrow failure. Recent studies have reported that the upregulation of Th17 and the downregulation of T regulatory cells in acquired aplastic anemia patients. However, what leads to this alteration in T cells remains unclear. Recently, it has shown that microRNA, which are short, non-coding RNAs, have been identified to be involved in the post-transcriptional regulation of T cells and their differentiation in the physiological as well as pathological conditions. This is a pictorial representation which are showing the role of microRNA in orchestrating the T cell functions. Here we can see the microRNA green in colors are upregulated, while those red in colors are downregulated when T cell subsets are formed. We already know that T cell repertoire is altered in aplastic anemia, so we speculate that the microRNA involved in the regulation of these T cells are also altered. However, this aspect has been poorly explored in acquired aplastic anemia. These are some of the pioneer studies show that the dysregulation of microRNAs in acquired aplastic anemia. A similar work was carried out in our lab, and we found that the upregulation of miR-155, miR-146, and miR-150, and the downregulation of miR-126 and miR-145 in the bone marrow mononuclear cells of acquired aplastic anemia patients. So with this background, our aim is to study the alteration in T cell subset in acquired aplastic anemia patients and evaluate the expression of microRNA specifically involved in the dysregulation of T cell subsets in acquired aplastic anemia patients, and also evaluate the expression of microRNA in follow-up patients. The study includes peripheral blood sample from 30 acquired aplastic anemia patients, 20 control, and 5 follow-up patients. T cell subset analysis was done by flow cytometers, and the expression analysis of microRNA was done by qRT-PCR, as shown in schematics. Now coming to the results, here we can see the percentage of CD3 positive T cells are significantly high in acquired aplastic anemia patients, but there is no changes in CD4 and CD8 positive T cells in acquired aplastic anemia patients as well as healthy donors. Further, we found the upregulation of Th17 and the downregulation of T regulatory cells in acquired aplastic anemia patients. Further, the expression analysis of microRNA, we found the upregulation of miR-155, miR-146, and miR-17, and the downregulation of miR-181, miR-326, miR-16, and miR-126. So this data highlights that the microRNA are altered in acquired aplastic anemia patients. Further, the expression analysis of microRNA in follow-up patient, we found the restoration expression of miR-155, miR-181, and miR-17, but there is no difference in the expression of miR-126, miR-16, and miR-326. This highlights the prognostic relevance of microRNA in acquired aplastic anemia patients. So in conclusion, our findings highlight that there is an alteration in T cell subsets in acquired aplastic anemia patients, Although literature review has shown controversial result for this imbalance in Th17 and Treg cells in acquired aplastic anemia. However, our results show the upregulation of Th17 and the downregulation of Treg cells in acquired aplastic anemia patients. Further, the expression analysis of microRNA, and we found the upregulation of MIR146, MIR155, and MIR17, and the downregulation of MIR16, MIR181, and MIR326. So these microRNA involved in the development of Th17 and T regulatory cells. Here we see the microRNA 155 interact with ROR gamma, which involved in the development of Th17 cells, and MIR16 interact to FOXP3, which involved in development of T regulatory cells. So this confirmed that microRNA are involved in deregulation of T cell subsets in acquired aplastic anemia, and thus possibly contribute toward the disease pathobiology. However, large cohort study is needed to identify potential prognostic relevance of these microRNA. Moreover, in vitro and target studies in enriched T cell population is essential to confirm our findings. Thank you.